Thank you, Mary. Thank you for giving me the opportunity to show the last work we have done in the translational research in our hospital and research institute talking about DARS2 gene related to axonal Charcot-Marie-Tooth. This is a very collaborative effort doing in our laboratory and also the neuromuscular unit in our hospital, but also with colleagues from Marseille, that is located in Nathalie Bonello, that has to be Nathalie Bonello Palot, and also the relationship with the colleagues from the University of Tartu in Estonia. So it's possible to have here the, well, no. No what?
No pointer.
No pointer at all, okay. Forward here. Okay, thank you. So the idea is to show you two topics, very relevant now, not only talking about Charcot-Marie-Tooth, but also these 2 points that is relevant now in clinical genetics and many of the aspects of medicine today. Now we're talking about a new CMT disease, but it's really the amplification of the clinical disease spectrum related with this DARS2 gene that is happening a lot now in clinical work, but also thinking about that the amino acid tRNA synthetase, in this case mitochondria, also related to Charcot-Marie-Tooth neuropathy, axonal neuropathy. So this is the families we have here on the top, on the right, that we had the 3 families, one from our hospital to CIPS, 2 other from the Marseille, that is also to CIPS, and 1 isolated case from Estonia. Then all these patients has axonal neuropathy. You have here all the clinical picture, the table showing the major aspects. They have 2 mutations in the DARS2 genes. So this is a Biallelic situation in a autosomal recessive inheritance. And on the right, you have the picture regarding to the first patient A1 in our clinics. The other E and F is belonging to the patient from Marseille, and also the muscle RNA imaging from the patient from Estonia. So what we found here is 2 mutations in the family A from St. John de Déu in Barcelona. Both are 2 missense mutation, one affecting serine 238 to phenylalanine, the other one affecting the arginine 336, changing to cysteine. And in this case, you can see it's not possible to use the pointer. It's a problem, but because we performed the analysis in the Escherichia coli homolog of the DARS2, and we can explain, based in this in silico modeling, that both missense mutations that originally were described as a variant of unknown significance became probably likely pathogenic, and then we performed later on some functional studies, several functional studies, to demonstrate that really they were pathogenic variants. Similarly, we did in the family from Marseille. They have also, the patient have the same mutation on the serine 238, and also a French mutation that is pathogenic itself. And the patient from family C was the patient that had one variant involving the splice site in the exon 5, and also missense mutation, the proline for 503 changing to leucine. In these cases, we have a variant that were considered pathogenic or likely pathogenic. In the case of the splicing site variant, this splicing site variant induced the exon skipping of the exon 5. So as a whole, we have 3 families, 5 patients with an axonal phenotype having mutations in DARS2. So this is now the idea that DARS2 is a gene related to another clinical phenotype that is a leukoencephalopathy associated with involving brain stem and also spinal cord, and associated with increased lactate acid and The phenotype is strictly in these patients usually affect the central nervous system. Some of the patients develop later on axonal neuropathy, but it's in the first clinical expression, and in our case, the first expression has been and the only expression has been the axonal neuropathy, except in patients from Estonia, C1, that has also intellectual disability, but in a different complete phenotype that the leukoencephalopathy, that because it's not involving the white system. So we performed these experiments trying to demonstrate that the patients, in our case, the patients from Barcelona, the A1 and A2, we work with fibroblasts with these patients, and we demonstrate that there was some changes from the functional point of view. There was a reduction in the amount of protein in both A1 and A2 patients from the family A, but also we could observe that there was, later on, there was a reduction in the fluorescence intensity in these fibroblasts. And from the patient from Estonia, we performed this experiment, was performed in Estonia, that the aminoacylation activity was reduced in the HEC cells which express the variants of the patient. The missense proline 503 variant. We performed also other experiments to demonstrate that there was changes in the lab. You can say the colocalization of DARS2 with TOM20, that is a marker of the out mitochondrial membrane, and we observed that there was a reduction in this interaction, in the juxtaposition between mitochondria and DARS2. That could be because there is a reduction on the DARS2 protein, that could be, but also we perform experiment regarding to how is the mitochondrial dynamics or the mitochondrial morphology that is changing also regarding to the cytosol occupied by mitochondria and also the fragmentation of mitochondria. We have not performed more in-depth studies for the dynamics point of view, but it seems that there is a reduction or abnormalities of the mitochondrial network in these cases. And also we perform experimental experiments regarding to the respiratory chain, the electronic chain in the mitochondria, in a membrane mitochondria. When we observe on the left, we observe in the patient in the fibroblasts from patient A1, we observe an abnormality in the, what we really observe in these experiments is that the oxygen consumption was almost normal, except when was induced the maximal respiration. That is you can see between F and rotenone antinomycin, we can see that is a difference between the control and the fibroblast from the patient. When we induce the maximum respiration, there is an abnormal response from the patient fibroblasts. This is a different situation you have on the right. They have the results coming from the paper by Wan et al., published this year, regarding how is the behavior of the respiratory chain that is really completely abnormal. So it's a difference between both cases, our patient and the patient from human. And we postulate that really in the case of our patient that has only axonal Charcot-Marie-Tooth phenotype is related, could be related because the general environment that could be stressing or not cells with a specific situation. So they maybe explain why both type of patients with different mutations in the same gene could have different phenotypic and pathophysiological response. So the general idea coming from these 3 families, 5 patients, is that there is a tRNA synthesis or tRNA binding alteration that is happening in the mitochondrial ribosomes. We can observe for some patients also the DARS expression that is reduced and also an altered mitochondrial network and function from the respiratory point of view. So this could be relevant, and also could be there is a enzymatic reduction in enzymatic activity. So in this case, it's possible that 2 mutations shared by the family from Barcelona and from Marseille, that is the serine 238 phenyl, and it could be a hypomorphic variant that, along with a deleterious advancement, could generate axonal CMT. And in the case of patients with the leukoencephalopathy maybe have two deleterious variants. One of them used to be affecting a splicing at the exon, at the intron 1, between exon 1 and exon 2, and then could be one of the possible explanations from the genetic point of view, the two different phenotypes and two different pathophysiology. So this is the idea that we try to show you two aspects of the real situation of CMT pathophysiology, but it's happening in another aspect of medicine, that is the phenotypic pleiotropy, in this case based in DARS2 mutations, generating a peripheral nerve system pathology or central nerve system disease, that is the leukoencephalopathy associated with increased lactic acid. And then also to incorporate the mitochondrial amino acid tRNA synthesis into the pathophysiology of and the clinical phenotype of the Charcot-Marie-Tooth in general and axonal Charcot-Marie-Tooth in particular. So thank you very much. This is the work has been done, has been the part of the thesis of Berta Esteve, that is our colleague that is on the right. And also Lara Cantarero has contributed in the respiratory experiments, and Jana Henike by addressing with me all this work. And also our colleagues from the neuromuscular unit, especially Dani Natera, that also our colleagues from the Turku and Marseille University and other places also. Thank you very much.
Thank you very much, Paco. I'm just going to ask a quick question and then David. So they're very interesting, and I know my memory from the paper is that two of your siblings were in their 50s. Have you done spectroscopy? Because I've had a patient who was diagnosed as P.K. Thomas as having CMT2 in his teenage years, then developed extensive planters. And it wasn't until his 30s he developed a CNS phenotype with abnormal MRI spectroscopy and MRI, and he has DARS2s. So MRI spectroscopy is more sensitive at picking up the lactic elevation. Yes,
in our case it has been measured, but I mean, well.
So what I mean is you don't know they might develop a CNS phenotype.
Well, I don't know, not exactly. But I think this is, well, it's true, what do you mean, yes. Yes, I don't know.
It's very interesting, the spectrum.
Yes, very interesting.
Yes. A similar question, because I saw a similar patient with sensory ataxia, motor involvement, no evidence of CNS involvement, but the MRI was clearly abnormal as seen in the... So the question is, was MRI completely abnormal? Because this
I didn't saw this, but it's completely normal. Yeah. The only different thing is in the patient from DARS2, the patient, I mean, is still a pediatric patient with some moderate intellectual disability. There is a thin corpus callosum, but there is not a wide...
no white matter changes in the cerebellum.
It's completely normal. And the two patients from Barcelona, because we have not the MRI from the patient from Marseille, they are completely normal.
Okay,
they are now 21, 23 years old. So, yes.
So clearly should be. We have it on our panel now, clearly for the younger people that might just like CMT. Thank you very much.
Thank you.
Thank you.